Good afternoon, everyone. My name is Wendy, and today I want to tell you a story about a case of mistaken identity. So, in this era of deep metagenomic sequencing, we are finding thousands of novel viruses in human samples, but just because a virus is in you doesn't mean that it's infecting you, right? Today I present Alphapartitivirus viator, a novel virus we found in humans that turned out to be just a passenger on a journey through the digestive tract. So, our story begins with a major challenge in modern virology, also known as viral dark matter. When we sequence a human gut sample, we don't just see human viruses, we see everything that person ate, touched, or breathed, and we frequently detect members of the Partitiviridae family in human stool. These are classically defined as plant or fungal viruses that cause persistent, non lethal infections in their hosts. Their presence in human clinical datasets, ranging from diabetes studies to infant cohorts, raises a critical question. Are these viruses adapting to jump into mammals, or is this just noise in the data? So we focused on a specific novel virus, SOTU U170297, which we found across multiple unrelated human datasets, and this led to our central research question. Is this an emergent zoonotic pathogen or simply an environmental contaminant? And based on its taxonomic classification, we hypothesized that this is just a dietary hitchhiker, a virus entering the system via food. If this is true, we predicted two things. First, its ecology should mirror food consumption patterns, not infection clusters. And second, its genetic dialect, its codon usage, should still look like a plant virus, not a human one. So to test this, we used different approaches. So first, we performed a digital ecology mining using Serratus tools and the SRA and different tools from the NIH, just to trace every location this virus has ever been seen. And we looked specifically at metadata distinctions like wild versus captive animals. And then second, we use AlphaFold 3 to solve the 3D structure of the viral polymerase to confirm it was indeed a partitivvirus. And finally, and most importantly, we applied codon usage forensics. And so we calculated the codon adaptation index, also known as CAI, and this effectively asks, is this virus speaking human, plant, or yeast? So, our ecological analysis revealed a striking pattern we call the captivity effect. We found the virus in humans in Europe and Africa, but what was interesting is that it also was found in primates. In a study of Macaca fascicularis, the virus was completely absent in wild macaques foraging in the forest, but it was found exclusively in a captive macaque, which was also fed a diet of chow and fresh produce. And this distribution strongly suggested that the virus is not being passed from monkey to monkey, but is being introduced by a shared agricultural supply chain. But the molecular evidence is even stronger. This plot shows the CAI of the virus against four potential hosts, and if this virus were infecting humans, we would expect its genetic sequence to have evolved to match our own cellular machinery. But instead, we see the exact opposite. So the virus is statistically maladapted to the human genome. However, it shows a massive, highly significant adaptation to the plant model and also yeast, with z-scores indicating strong evolutionary pressure. And this is molecular proof that this virus evolved to replicate in a plant or fungal cell, not a mammalian one. So, in conclusion, our virus is a viator, a traveler, and it's a harmless dietary hitchhiker, likely ingested with produce or grain products, and it serves as a critical reminder for virology that high throughput sequencing is incredibly sensitive, but it requires biological context, and we must rigorously distinguish between active infection and environmental infiltration to prevent any misdiagnosis and better understand the true dynamics of the human virome. These are my references. Thank you for listening to my presentation.